Hello students, in this video we're going to go through topic 3 of these notes. In topic 3 we're going to look at how DNA replicates. So in the first two topics we talked about how, we talked about some experiments that led to proving that DNA is the genetic material, and we talked about how they very quickly figured out the three-dimensional structure of DNA. And so the next big task after learning about or knowing or finding out that DNA is the genetic material and learning its three-dimensional structure, the next big question was, how does it replicate? Because if it is the genetic material, DNA, if it is the genetic material, that means that every time a cell divides and makes new cells, each one is going to need a complete copy of that genetic material, the DNA. And so they wanted to know how does this molecule of DNA replicate to make sure that it's always passing on that information from each generation of cells. And so conveniently, with Watson and Crick's discovery of the structure of DNA, it became kind of obvious of how this might work, of how DNA might replicate. Since DNA is made of two strands that are complementary to each other, it's double-stranded, and each of those strands there's complementary base pairing that takes place. Well, the logical guess on how DNA replicates is that those two strands would somehow separate, and then you could use each of those strands as a template to build the missing strand because of these base pairing rules that A has to go with T and C has to go with G and so forth. And so just having one of these strands would be enough information to build a new strand, or having this other strand would be enough information to build the other missing strand. And so you could end up with a molecule now becoming two molecules of DNA, or being able to have the information to build those new strands of DNA. And so there's three ways that this could have technically played out, or there's these three competing models of how this might play out. There's the conservative model, the semi-conservative model, and dispersive model. One of these is correct, which we'll go over in a second, but I just want to show you guys each of these three ideas real quick. Starting with the conservative model, according to this idea was that the two original parent, we'll call them the parent strands, the two parent strands of DNA would separate, and then they would be used to build two new strands, and those two new strands would then end up together, and the two original strands would go back together. And so you'd end up with these two molecules of DNA, one of those molecules made with brand new DNA, and one of those molecules still has all of the original DNA, both strands, or the DNA that we started with. And then that could happen again, and then those two strands could separate and be used to build two new strands that come together, but then those two strands go back together. Same thing here. And so each generation of DNA replication, you'd always have those two very original strands end up together, right? So we can even track them. In this picture, they're color coding the two original strands of DNA that we started with before you replicated the first time, before you replicated the second time. You can see that they're always ending up back together because they're going to separate and be used, that information to be used to build two new strands, but those two new strands would end up together, and those two original strands would come back together. And so after it replicates once, you'd have DNA that's 100% new and 100% the original DNA. And then after it replicates again, you'd still have a bunch of DNA molecules that have new DNA that was made in the last replication or in this replication. But there'd still be molecules that have 100% the original DNA. Now in the semi-conservative model, you can see how this is a little bit different. This model, according to this model, the two strands of DNA would separate, but they would separate permanently. And then each of those strands would be used to build a new complementary strand, a new strand of DNA that they would then stick with. And so you could see that happening here. Those two strands separate, and then they're being used to build two new strands. But those new strands will pair and stay with one of the original strands. And then you can see that happening again. Those two strands will separate and then be used to build a brand new strand. And in this picture, again, you can see those two original strands of DNA, the dark blue ones, where they end up after it replicates once and after it replicates twice. And all this light blue DNA, that's DNA that was made, that's new DNA that was made in the first replication of their second replication. And so after the first replication, you have this DNA that's 50%, these DNA molecules that are 50% the original DNA and 50% the new DNA. But after it replicates a second time, you now have molecules that are still 50% have one, like they have one of those strands that's the original DNA, so they're 50% new DNA, 50% old DNA. But you also have some strands that are now don't have any of the original DNA. These strands don't have any of those original strands of DNA because one of those strands was made last time and one of them was made this time. So some of that DNA all of that DNA is new, either made in the first replication or the second replication. And then the dispersive model, this one's a little bit messy, but basically there's going to be chunks or pieces of each strand being used in each of the 2 new daughter DNA molecules. And so we're going to take pieces of this strand and the other strand and this strand and the other strand, and then we're going to fill in the missing pieces on both strands with new DNA. And so this original parent DNA is being dispersed 50-50 between each new strand and these new molecules, and then it's being dispersed again. And so after it replicates the first time, you have DNA molecules that are 50% the original DNA, about 50% of the DNA, original DNA is in here, and about 50% is here, and 50% is new and new. But then after the second time it replicates, you now have DNA molecules, like if you kept dispersing all of this DNA evenly between that was in these 2 strands to these new molecules that are forming, you now have DNA, these DNA molecules, each of them have about 25% of that very original DNA that we started with after it was dispersed 50-50 and then dispersed a little bit more. And then each time you have to kind of fill in the missing pieces with new DNA is what that's showing here. So those are the 3 competing models of how DNA replicates. Now there was an experiment that was put together to figure out which one of these is actually correct. And that is the Michelson-Stahl experiment. And in this experiment, it's kind of confusing at first, but I'll try my best to make this make sense. What they're going to do is they're going to start with DNA molecules that contain a certain isotope of nitrogen. So DNA has a lot of nitrogen inside of it, and they're going to use a specific isotope of nitrogen that's heavier than the normal isotope of nitrogen. So this is nitrogen 15, that has a mass of 15, and then there's nitrogen with a mass of 14. So those are 2 isotopes of nitrogen. And so we're going to call this the heavy isotope, the heavy nitrogen, and this is the light nitrogen. And so what they're going to do is they're going to start with DNA molecules that are made with 100% this heavy nitrogen. So all the nitrogen in this DNA that we're starting with is using this heavy isotope of nitrogen. So these DNA molecules would be like literally heavier than usual. Okay, like they're going to have more mass, way more than DNA that's made of this lighter version of nitrogen. So they're going to start with DNA molecules that have this heavy nitrogen, okay? And then what they're going to do is they're going to take that DNA, those DNA molecules that have the heavy nitrogen, and they're going to let those DNA molecules replicate, but they're going to let them replicate in a medium that only has the light, normal nitrogen. And so if they replicate, any new DNA that's being made is going to have to use the lighter version of nitrogen. But we'll be able to keep track of what was the original DNA, because the original DNA will always have the heavy nitrogen, but any new DNA from that point on, when it replicates once or twice or 5 times or whatever, any new DNA from that point on is going to use the lighter nitrogen. And then here they're kind of color coding it for you. And so what they do is before they let it replicate, they're going to take this solution of DNA, basically just imagine a bunch of DNA molecules in here that all have the heavy nitrogen. So there's DNA molecules in here that contain 100% the heavy nitrogen. And they pour that into a little tiny test tube and they centrifuge it. And by centrifuging it, it's going to cause all of these molecules to clump together based off of their mass or their density. And so if the molecules have the same mass or density, they're going to end up clumping together in the same spot on this test tube once it spins really, really fast. And so at first, when they do that, in the test tube, there's a single band that forms near the kind of closer to the bottom of the test tube. And that's because all the DNA molecules all weigh the same at the very beginning of this experiment. They all weigh the same and they all contain the heavy nitrogen. And so when you spin that in a test tube, they're all going to clump together kind of near the bottom of this test tube because they're heavier, okay? And then after they're going to let those DNA molecules replicate once. And again, they're going to let it replicate in a medium containing the light nitrogen. So any new DNA from this point on is going to have to use the light nitrogen. And after they let those DNA molecules replicate once, so all those DNA molecules, they can replicate one time, they're then going to take that solution then of all this DNA that's been replicated once, and they're going to pour it back into a little tiny test tube, and they're going to centrifuge it, and all those DNA molecules are going to clump together based off of their mass. And what they found is that they all clumped together in the same spot, which means all those DNA molecules weighed the same, but they clumped at a slightly higher spot than the first time, which means that those DNA molecules all weigh the same, but now they're lighter than they used to be. And so that right there told them that one of these models of DNA replication is incorrect. after it replicated once. Once they got that data after it replicated once, it actually proved that it couldn't be the conservative model. Because if it was the conservative model, you would have this original DNA that had heavy nitrogen. So imagine the dark blue represents the heavy nitrogen. And those two strands would have stayed together in the DNA molecules. And so you would still have DNA molecules that have 100% the heavy DNA. But then at this point, you would have now new DNA molecules that have that are made of new DNA, which would have to be the light nitrogen. So imagine this light blue represents the light isotope of nitrogen. And so these molecules would be lighter than these molecules. And so you should have had DNA clump in two spots in the test tube. You should have had DNA clumping towards the bottom where it was originally, but now you'll have some DNA clumping higher up. There'd be two bands of DNA that form, which is not what happened in their experiment. There was only one band of DNA forming. And then what they did is they let it replicate a second time. So they let it go through 2 rounds of replication. So they took this DNA that had 100% the heavy nitrogen. They let it replicate once in the presence of the light nitrogen. So any new DNA has to use the light nitrogen. And then they let it replicate again. Again, any new DNA is going to have to use the light nitrogen. So the only DNA that's going to have the heavy nitrogen is the original DNA molecules that you started with, the original strands that you started with. And once they did that twice, and they let it replicate twice, they then poured it in a little tiny test tube and they centrifuged it. And what they found is that the DNA clumped into 2 spots or 2 bands in this test tube. Some of that DNA clumped in the middle like last time. And then some of it now, though, there was a second band of DNA clumping higher up. And so what that told them is after it replicated twice, you now have DNA molecules that weigh different amounts. Some of the DNA molecules are lighter than other DNA molecules. And some of them were as heavy as they were last time, but now some of them are lighter than they were last time. And that actually is what proved that it had to be the semi-conservative model. Because if you look here, when those two, after it replicates once, you're going to have DNA molecules that weigh the same. Each of these DNA molecules is going to have 50% the heavy nitrogen and 50% the light nitrogen because it's new DNA that's being made. And that would form a single band in the test tube, which is what happened here. There was a single band that formed after it replicated once. And then after it replicates twice, again, only using the light nitrogen to make any new DNA, you're going to now have DNA molecules that are like this, where some of them still contain one of those original heavy nitrogen strands, like this one and this one, but the other strand is the light nitrogen. So these guys are about 50% heavy, 50% light. But then now there's going to be molecules of DNA that don't have any of the original DNA. Like they have a new strand that was made last time with the light nitrogen and a new strand that was made this time when it replicated a second time. That's also made of the light nitrogen. So now you're going to have DNA molecules that are 100% made of the light nitrogen. They don't have any of the original DNA that we started the experiment with. But you're still going to have some DNA molecules that have a strand that contains that original heavy nitrogen DNA. And so this would be molecules that weigh different amounts. These DNA molecules here and here would be 50% heavy, 50% light, kind of like a medium weight. But then these guys right here are going to be way different. They're going to weigh they're going to be lighter than these DNA molecules. They're not going to weigh the same. And so you're going to have 2 types of DNA molecules, some that are lighter than others, which is what's happening exactly right here after it replicated twice. Now, if this was the dispersive model, after it replicates once, they would be 50-50 heavy light, which would be like the same band. So it'd be like a single band like right here. But after it replicated twice, you would now have each DNA molecule would still weigh about the same. Each of them would still have 25% of that original heavy DNA and be 75% new DNA by the time it replicated twice if it was the dispersive model. So after it replicated twice, these DNA molecules would have clumped together because they all would have weighed about the same, 25% that heavy original nitrogen, but 75% this new DNA. And it would be lighter than it was the last time, which was lighter than it was the first time. But that's not what happened in their results. After it replicated twice, there was 2 distinct bands. And so these DNA molecules no longer weighed the same. And so that's what showed that it was the semi-conservative model of DNA replication. This picture might even be better. Maybe I should have walked you guys through this picture, but it's showing you guys the same thing, that you're starting with this DNA, these DNA molecules that have the heavy nitrogen, they form a band at the bottom, but after it replicates once, now the DNA ends up in the middle, and that's because those DNA molecules have half of the original heavy DNA and then half of this new light DNA, and they each weigh the same. They're 50-50 heavy and light, or new and old DNA, 50-50 new and old DNA. And so they clump in the middle. They're a little bit lighter than they were the first time. And then after it replicates twice, though, you're going to now have some DNA molecules that have none of that original DNA. But you'll still have some DNA molecules that have some of that original DNA, like one of these original heavy strands. And so that's now going to form two bands, some in the middle where they were last time, because that's the DNA that was 50-50, heavy and light. But now you're going to have some even higher up on the test tube after it's centrifuged, which is all of the DNA molecules that contain 100% newer DNA. That's the lighter nitrogen. They have none of the original heavy nitrogen inside of them. So I hope that kind of makes sense. We'll talk more of that in class because it is kind of confusing. It's easier to kind of show you guys in class with like a whiteboard what's going on, but that's the gist of it. Just know that the experiment showed it's the semi-conservative model of DNA replication. And so then the remainder of these slides are going to talk about the actual mechanism of how DNA replicates on a molecular level involving enzymes and proteins. And so I'll walk you guys through that. So DNA replication, if you have a DNA strand, it's going to start at locations called origin of replication, or we abbreviate ORI for short, or ORI for short. And that's a spot on a DNA molecule where we're going to start replicating the DNA. And so that's where the two strands are going to first separate. And then we're going to start copying those two strands in both directions to make the new, so that we end up with two new DNA molecules. So you can see here that we have multiple spots on a DNA molecule that we're starting replication. We're going to open up the DNA and start copying in both directions. And then by the end of it, you're going to now have two DNA molecules. Each of them having one strand of the original DNA. But now each of them now has a newly built DNA strand for both of them. And so for eukaryotes, we actually have on your guys' chromosomes and a eukaryotic chromosome, which is very, very big. Like it can be billions of nucleotides long. There's many origins of replication. So when your chromosomes are copying, your DNA is replicating, it's going to start at many different spots on your chromosome so that it can happen a lot faster. In prokaryotes, though, just a difference between us and prokaryotes. Prokaryotes, they have a circular chromosome, and they only have one origin of replication. So replication starts at one spot on that chromosome and then goes both ways until the whole chromosome is replicated. So these are called replication bubbles, where the DNA is opening up and we're starting to do DNA replication in both directions. And then here's a replication fork, where you can actually see the DNA starting to be made as it's moving along and opening up this DNA more. So we're going to kind of focus in and zoom in here on what's happening at a replication fork. How is this new DNA being made on both of these strands? And so this actually involves lots of different enzymes. A lot of these enzymes and proteins involved, they're catalyzing different reactions and doing different things, working together to accomplish the replication of this DNA molecule. A lot of them are clumped and bonded together to form a giant DNA replication complex. But in the pictures I show you, they'll show them kind of like independent proteins, but just know they're actually kind of all together, forming this superstructure here. But there's 5 main proteins that you guys need to know and what they're doing during DNA replication. So I'm going to walk through those right now. So first of all, there's helicase. Helicase is an enzyme, a protein, who actually is the one unzipping the DNA. So he's separating the hydrogen bonds between these nitrogenous bases and allowing these two strands to separate. So now we have our two strands that we can build off of. And then as he's unzipping, though, because DNA is a double helix, because DNA have two strands that are twisted around each other, when you start unzipping that, it creates something called supercoiling ahead of helicase. So as he's unzipping ahead of this replication fork, the DNA gets really, really tightly coiled as you start unzipping it. Imagine two strings twisted around each other, and then you start pulling them apart. It starts to get really, really twisted up ahead. And so that can damage the DNA. So there's this other protein or enzyme called topoisomerase. He's an enzyme who will be ahead of helicase, ahead of the replication fork, and he's going to relieve the supercoiling. So he can actually break the DNA strands and allow them to unwind a little bit so that they don't get too coiled and end up damaging the DNA. So there's two of the enzymes you guys need to know. Helicase unzips it, topoisomerase relieves the supercoiling ahead of the replication fork as helicase is unzipping the DNA. And then you have DNA polymerase. DNA polymerase is the enzyme who's actually building the new strand. So he's synthesizing the new strand of DNA using the original strand as a template. So this is the template that he's building off of, complementary to it. And so he's adding these new nucleotides together, as A's and T's and G's and C's, as he's reading this template strand and putting these nucleotides and bonding these nucleotides together in this new strand of DNA. So he's doing that here and he's doing that down here. The thing about DNA polymerase, though, there's a couple important things you guys need to know. He can only synthesize the new DNA in the 5' to 3' direction. So if you guys remember, DNA is anti-parallel. And so there's a 5' end and a 3' end here. So you can see up here, he's building the new strand starting on the 5' end of this new strand, and he's building towards the 3' end. So he can only add new nucleotides to the 3' end. If you guys remember me pointing this out many times this year, when you add new nucleotides, you can only add to the 3' end. That's because this enzyme, DNA polymerase, he can only add new nucleotides and bind them to the 3' end of a pre-existing strand. So here's our strand that we're building. He can only build off this 3' end. He can't build off the 5' end. So we say that he builds 5' to 3' in that direction, which as he's doing that, he's reading the template in the 3' to 5' direction. So we say, so if you listen carefully here, he's reading, here's the template strand. He's reading that strand in the 3' to 5' direction as he's building the new strand in the 5' to 3' direction, because it's going to be anti-parallel. And so that's something really important you guys need to know about DNA polymerase, okay? He's very specific in how he's putting together these new nucleotides. Now that actually creates a problem, because on one of those strands that works out pretty conveniently where he's going to be moving in this direction, which is the same direction that DNA is unzipping, like helicase, who's not being shown here. He's unzipping the DNA and we're moving to the left in this picture, right? And he's also moving to the left. But on the other strand, DNA polymerase is going to have to work in the opposite direction. Because DNA is anti-parallel, that other strand is running in a different 5' to 3' direction. And since he can only build in the 5' to 3' direction, on this bottom strand, he's actually going to have to build it to the right. So he's going to have to build it towards the right, even though everyone's headed to the left, like we're trying to undo the DNA this way. And so that causes, on one of these strands, the DNA to be built in tiny fragments. So he's going to build a piece going to the right because he has to go to the right. He has to build 5' to 3', and then he's going to let go and then come back and do another section, 5' to 3' to the right, and then another section and another section. So he's going to have to build one of these strands in pieces, whereas the other strand is just continuous. So the strand that's being built continuously, because it's going the same direction as the replication fork, is called the leading strand. And the one that's being built discontinuously, meaning like we have to build a piece, then come back and build another piece, and then come back and build another piece, that's called the lagging strand. It's called the lagging strand because it's going to take a lot more time and effort to get that strand made. And so here you can kind of see a better picture where, because DNA is anti-parallel and we have to build in the 5' to 3' direction, excuse me, on one of those strands, that's going to be in the same direction as we're unzipping the DNA, which that means that we can just continuously build off the 3' end in this direction. But in the other strand, since it has to be built 5' to 3' and the other strand is going the opposite direction, we're going to have to be building it in the opposite direction away from the replication fork, 5' to 3', just like this one's being built 5' to 3', but that's going to be going to the right on this strand. And so we're going to have to build a piece and then come back and build another piece and come back and build another piece. So that's what I mean by discontinuous. That's the lagging strand. And these fragments that we end up with of DNA being made on the lagging strand are called Okazaki fragments, named after the scientist who discovered these fragments and figured out that there's a leading and lagging strand. And then soon those Okazaki fragments, those pieces of DNA being built in segments here on the lagging strand, they'll be joined together by an enzyme called ligase. So ligase will make the final bonds between them on the sugar phosphate backbone. He'll join them together, those nucleotides together, so that the strand is perfectly continuous, and you would never know that there was fragments there because there's this other enzyme called ligase who's going to join those fragments together by basically making this last missing bond between those fragments, which we're going to have to do that a lot on the lagging strand. And then another thing that you guys need to know is, or another enzyme you guys need to know is an enzyme called primase. So primase is an enzyme who's going to make a short fragment of RNA complementary to the DNA template. So it's going to build a short piece of RNA, and we call that a primer. And the reason that's being made is because DNA polymerase can only build off of an existing strand. And so basically DNA polymerase has to start with a strand that already exists, and then he can build off of it with more and more DNA. And so DNA polymerase will always start with an RNA primer, this piece that was put here, and he'll start building off of it with DNA. Which is what's going on here. He's building the new strand, but he always has to start with a primer. And that primer is made by this enzyme called primase, and it's actually made of RNA nucleotides. So in these primers, which are made of RNA nucleotides, they're going to have some uracil and not thymine, and they're going to have the sugar ribose. So now we're going to have some pieces of RNA in this new DNA that's being made. But that's because DNA polymerase, he can't start from scratch. He can only build off the 3' end of a pre-existing strand, which is the whole point of putting this primer here by this guy, primase, so that he can build off of it. Now on the leading strand, you're only going to need one of those. And once you have one primer, you can just keep going and going and going and build off of it. But on the lagging strand, as you guys can imagine, you're going to need lots of primers. Every time that DNA polymerase makes a short piece, when he comes back to make another piece, there has to be a primer there so that he can build off of it. And so on the lagging strand, you're going to see lots of primers there made by primase. He's going to put these short pieces of RNA, and he's going to have them ready so that DNA polymerase can then come and build off of it to make the new DNA. And he's going to have to do that over and over and over again as we continue to unzip the DNA. And then sooner or later, those primers, which are made of RNA, they won't stay there. They'll get replaced at some point by another enzyme. There's actually another type of DNA polymerase, not the same. There's different types of DNA polymerases. There's another type of DNA polymerase who actually will come back and remove the RNA primers and replace them with DNA nucleotides, so that so by the end of all of this, there will not be any RNA left in your DNA. So as you're replicating your DNA, you're putting on these short little RNA pieces, and then this DNA polymerase will come back and remove those RNA pieces and replace them with DNA. And then that's when ligase will come and make those final missing bonds between these fragments. And so this picture here is showing you the same thing. So here's a primer, DNA polymerase is building off of it. But then at some point, another DNA polymerase will come and replace those primers with DNA. And then there's still going to be one last bond missing in the sugar phosphate backbone between those fragments. And so DNA ligase makes that final bond. And then you have perfectly made new DNA. So the lagging strand is a lot of work. And it's all because of DNA polymerase having to build in the 5' to 3' direction, which on one of these strands is going to be inconvenient because he's going to be going the opposite way, and he's going to have to do it in short pieces and then come back and build another short piece and then come back and build another short piece. And then lastly on these slides, it talks about the mechanisms in place to make sure that when your guys' DNA is replicating, that there's not a lot of mistakes. So during DNA replication, this is actually the biggest source of mutations in organisms and in cells. When your guys' DNA is replicating, your cells are taking billions of nucleotides. You have very long strands of DNA in your guys' chromosomes, and it's copying all of it. And it's very common, kind of common actually, that when the new DNA is being made, you end up with some mistakes in that newly made DNA. So DNA polymerase, he's not perfect as he's putting together the A's and the T's and the G's and the C's. Sometimes he messes up. But luckily, there's some mechanisms in place to kind of fix some of those mistakes. First of all, DNA polymerase has a proofreading mechanism. So if he does put the wrong nucleotide, when he's building the new strand and he puts the incorrect nucleotide, like let's say it's a T when it's supposed to be a G, usually he can actually, this protein can actually catch that he made a mistake. He can catch himself, and then he'll stop and then try again. He'll remove it and then put another one on. So it's kind of like as you guys are typing an essay, while you're typing the essay, you might find, like, as you're typing a sentence, you made a mistake or a typo, and then in that moment, you're fixing it. Like, so technically you did make a mistake, but you fixed it as you were going along. So same thing with DNA polymerase. He makes mistakes commonly, kind of, but he's catching a lot of them as he's making those mistakes. But then there's going to be some mistakes that he didn't catch. Just like when you guys write an essay, there's going to be some typos that you missed as you were writing it. And so you're going to end up sometimes with some incorrect bases in those new DNA molecules that are being made. And so there's other mechanisms, though, that involve other proteins. There's other proteins in your cells that will actually go check your newly replicated DNA to make sure that there's no mistakes. And if it finds mistakes, it will actually, there's proteins who actually cut out those mistakes, which will then signal DNA polymerase to come back and fix it. And so that kind of fixes those mistakes. So that's called mismatch repair, when there's a mismatch and then we cut it out later on and replace it and fix it. So by the end, because of these are two examples of some ways in which cells are fixing these mistakes during DNA replication. So by the end of it, after you replicate all your DNA, it's very, very accurate. But there's still, even after all of this, though, there will sometimes be mistakes left in your DNA, mutations that occur, even after we're fixing a lot of these mistakes. But very, very small amounts of mistakes had we not had these other mechanisms in place. Anyway, that's it for this video. Hopefully that kind of makes sense. So we'll talk a lot about DNA replication in class. So hopefully, as of right now, you got a good initial understanding of all of it. Anyway, thank you guys, and I'll see you later.